multiple times that, you know, the kidneys are responsible for excreting things, so getting rid of fluid, ions, items that we don't necessarily need, okay, from our bloodstream. So it's responsible for eliminating, but even more importantly, it is responsible for monitoring and regulating our blood pressure. And it does that by monitoring our blood volume. Because it's sensitive to water levels and ion levels, it's sensitive to volume of blood, which is directly related to blood pressure, okay? So if it's gonna work to maintain a proper balance of water and salts, acids, bases, what have you, ions in general, then it just lends itself nicely to being kind of a monitor for your blood pressure. Okay, and we'll talk about different scenarios that can happen with that. Okay, so as waste, okay, there are three main things that we excrete from our body. Urea is the first one, and that's usually from the breakdown of proteins. Okay, then you've got uric acid, which is the breakdown of nucleic acids, so DNA. And then we didn't really talk about this in the muscle section, but creatinine is another waste product, which is a kind of byproduct of a phosphate that is found in your muscle groups. Okay, so those three items are typical waste products that you will find in your urine. Okay? Okay, so basic anatomy, all right, kidneys we know already, retroperitoneal, so they're behind that peritoneal membrane. Each one's about the size of a bar of soap, just FYI. The right one is slightly inferior because of the liver, and we know that the adrenal glands live right on top of them. That's why they're called the suprarenal glands, okay? So nothing new there. As far as connective tissue goes, there's three layers of connective tissue that surround the kidneys, okay? And in lab, we learned one of them. The first one is the fibrous capsule, okay? So the fibrous capsule, if you're touching your kidney, okay, deep on the models, that one outer layer on that model is considered the fibrous capsule. Okay, so that's the deepest layer. Next to that, we have what's called perirenal fat capsule. Perirenal meaning around the kidneys. So this is going to be what kind of tissue? Adipose. Adipose connective tissue, and it's for cushion, it's for protection. Okay, so that's the next layer. So then the most superficial layer is called renal fascia. Okay, we have anterior renal fascia and posterior renal fascia. Anterior renal fascia anchors itself to the parietal peritoneum. Okay, so it provides structural support anteriorly to the kidney. Posteriorly, the renal fascia attaches to the musculature of the low back. So a little bit of the erector spinae fascia. Okay. And in dissecting the cat and looking at all musculature on the cat, we are well versed in what fascia is and where you find it. It's everywhere. Okay. Okay. So a lot of this is not new. Okay. Because it's a solid organ, outer cortex, inner medulla, we know that the medulla makes these structures called renal pyramids. Okay. Papilla is what the pyramids come to, okay, kind of medially on the organ, the little bumps. And then the papilla lead into these little channels called what? Minor calyces, right, or a minor calyx. Yeah. Now, renal columns are going to be that part of the cortex that kind of invaginates itself in between the pyramids. Okay, so that's a column. And then a lobe, that's probably the only new thing on here. A lobe, they put it in dotted lines right here in black, a renal lobe. So it's basically a renal pyramid and half of the column on either side. So the half of the column of cortex on either side and a pyramid is considered a renal lobe, okay? And we know the hilus is also called the hilum. It's where vessels enter and exit the organ. Okay, so as far as lobes go for this particular kidney, we're going to have 1, 2, 3, 4, 5, 6, 7 lobes. Okay. All right, nothing new here. Okay. So minor calyces, okay, are the channels that the papilla attach to. Minor calyces combine to form what? A major calyx, which then empties into the renal pelvis and out the ureter. Okay? So nothing new there. Okay, so blood flow we need to know. All right, we've identified all of this already. Okay, so we know that coming into the kidneys as an organ are the big renals, okay? The renal artery, the first branch off the renal artery is called what? Segmental artery, right? Is there a segmental vein? No. No, there's not, okay? So we go from segmental artery then to interlobar. So interlobar kind of aligns itself on either side of the renal pyramids, okay? Interlobar then makes an arc and forms the arcuate artery and vein. They all flow together, right? So the arcuates are nice because the arcuates create a boundary between the medulla and the cortex in the organ, okay? And then coming off the arcuate arteries are the cortical radiate arteries, which means radiating into the cortex, so that's kind of a nice name. Branching off of those cortical radiates you guys saw in the models in lab, now we start the afferent arteriole. And the afferent arteriole is going to feed a capillary bed called what? Called the glomerulus, right? So the afferent arteriole feeds the glomerulus, which we know is a capillary bed. It's unique in that it's both fed and drained by an arteriole, right? So coming out of the glomerulus is what vessel? The efferent arteriole, because efferent means away, right? So efferent arteriole. And then we don't immediately go into the venous system. We have a system of capillaries first that you guys identified. One in the cortex and one in the medulla. Which capillary bed is in the cortex? Peritubular capillaries. So the vasa recta is found in the medulla. Okay. So that blood then enters the capillary beds, which allows for continued reabsorption of whatever it is we need. Okay. We may still need ions, we may still need some water. So there's that capability still available to us. And then we'll go through the exact steps here in a minute. And then we enter the venous system, headed back to the heart. Okay? So there's multiple opportunities configured into this flow to allow for us to reabsorb water and ions if we need to at any point in time. Okay, so we need to know flow. All right, so again, these are not new. We know that the functional unit of the kidney is called the nephron. What's the functional unit of the lung? The alveolus or alveoli. What about the nervous system? The neuron, right? Yep, so each organ system has its own, and we're talking about the nephron here for the kidneys. So we've got the renal corpuscle, which includes the glomerulus and the capsule, and the capsular space is fine, capsular space inside, okay? And then we have part of the tubule. And the PCT stands for what? Proximal convoluted tubule. Then we have the loop of Henle, which is also called the nephron loop, where you have your descending limb and your ascending limb. And then what's the DCT? Distal convoluted tubule, right? And then we finally end in the collecting duct, which is not part of the nephron, okay, but it can attach to many nephrons, okay, as part of flow, all right? And we'll talk about that in a minute. Okay, so again, nothing really new here. This picture on the side looks just like one of the models in lab. Again, the arcuate vessels are really nice because they give you that boundary between medulla and cortex, okay? The collecting duct is what goes all the way down to the papilla. So the collecting duct is basically the last tube, okay, that fluid's gonna enter, so this is gonna be your last opportunity to reabsorb anything, get rid of it or add it before it exits the papilla. Because once it exits the papilla and enters a minor calyx, at that point, it's considered urine. Okay? It's not considered urine until then. So we'll talk about that. Okay, so we've got three main mechanisms to produce urine, okay? The first one is filtration, okay? And filtration is step one, okay? That's where fluid, namely plasma, okay, because it's a derivative of your bloodstream, blood plasma leaves the glomerulus. Okay, so it's gonna leave the capillaries. So filtrate, okay, once fluid leaves the glomerulus, okay, it is now out of your bloodstream proper, and it's considered filtrate. It's in the glomerular space, it's considered filtrate. It's not the same consistency of your blood, but it is a derivative of your plasma, okay? So you're gonna have lower protein, and you're also gonna have you're not gonna have cells in it. You're not gonna have white blood cells, red blood cells. You don't wanna filter those out. You still need those. Okay, so you're filtering out water, you're filtering out chloride, magnesium, sulfur, whatever other ions, okay? Sodium, okay, potassium, right? That's what you're filtering, okay? So that filtrate then from step one enters this tube. What is this tube called?
Proximal convoluted.
This is the proximal convoluted tubule, okay? Which is where reabsorption occurs, okay? When we're saying reabsorption means you already had it before, so it was once in your bloodstream, but you're taking it back again because you filtered it out, but you want it back. So you're reabsorbing it, okay? You're not taking it in for the first time. This is something that got filtered, but you want it back. So it's reabsorption, okay? 99% of what gets filtered is reabsorbed. 99%. Okay? And this occurs throughout the tubule, throughout the PCT, the loop, and the DCT, and the collecting duct. And we'll talk about what goes where and why. So what the kidney does, it's really interesting, it does this kind of all or none, right? It takes 99% out. So it's kind of like an interesting way to clean your room, if you will. So instead of just going into your room that's a hot disaster and like picking out a couple things, you're like, I don't need this, I don't need that, I don't need that. Instead, take everything out of your room and throw it in the hallway, and then pull back what you want. That's kind of what the kidney does. Takes it all out, and then it pulls back a majority of what it wants. Okay, so that's just the strategy that the kidney is using. Okay, then you also have an opportunity to secrete things from your bloodstream that you may not want. Okay, other ions, wastes, toxins, okay? There's an opportunity to remove that from your bloodstream because you have capillary beds that allow you to do this, okay? So, step two in the picture coming from the PCT is showing you reabsorption. It's going from the tube back into your bloodstream through your peritubular capillaries. So we here are in the cortex, okay? So that's why we identified capillaries both in the cortex and in the medulla, because you have multiple opportunities to throw things back into your bloodstream or take stuff out of your bloodstream, okay? So step three here is showing the arrow going from your bloodstream out into the PCT. That's secretion. That's your bloodstream saying, oh, I have too much of this, I don't need this, I'm gonna throw this into the PCT. So it gives your bloodstream ample opportunities to get rid of things along the way. It's not just filtration where you get rid of everything. Because again, 99% of it's thrown back in your bloodstream. So your bloodstream may have other items that it wants to get rid of. That wasn't necessarily part of the filtrate, so it can, because it can secrete them back into the PCT because of the peritubular capillaries. Okay. So things like toxins, drugs, antibiotics, other ions that you don't need. Okay. All right. Okay, so let's review again. It's always good to repeat ourselves. So the corpuscle, right? Glomerulus, capsule, and we can call it capsular space too, that's fine. It's all in one, okay? We know the glomerulus is capillaries. It's fenestrated capillaries. Fenestrations are what?
Holes.
Pores, right? Or holes, right? We had continuous, fenestrated, and what was the most leaky? Sinusoid. Okay, so we're middle of the road here. So we got pores, okay? We have an afferent going in, we have an efferent going out, okay? So because afferent is larger than efferent, what did we say that does to blood flow through the glomerulus? It slows it down so it gives time for filtration to happen. Because of that, the blood pressure within your glomeruli is very high, okay? Because that facilitates filtration, okay? Facilitates ions leaving. Okay. All right, so Bowman's capsule, or the glomerular capsule, has two layers, like a serous membrane. It has an outer parietal layer of simple squamous, because again, out of the capsule, is filtrate, and then a visceral layer, which consists of those podocytes that we talked about and identified in lab. So the podocytes that you guys identified on the model have a lot of little extensions coming off of them. And it serves as like another filter, if you will, for substances that want to come out of the glomerulus, okay? So the glomerulus has a special strategy to ensure that what gets filtered out should be filtered out. And things like red blood cells and white blood cells and platelets and other things don't, okay? Because we want to hold on to that. So we have what's called a filtration membrane, okay? What kind of membrane did we talk about in the respiratory system? The respiratory membrane, right? It's the same thing. So it's where all the magic happens, okay, in terms of filtrating. So you've got the fenestrated pores of the glomerulus, and then you have these fingers of the podocytes that creates like another screen, okay? And then in between those two layers, you have a basement membrane, or like a lamina propria, where both of them can attach to, okay? So the next slide basically gives you this rundown, okay? So you've got your pores here in your fenestrated glomerulus, fenestrated capillary. Here's your blood plasma, okay, because you're in the glomerulus. On this side, you have all of those podocytes that interdigitate, so they have spaces in between them. And then in between the two, you have this basement membrane, or lamina propria. Okay. Again, thin layer of epithelial tissue that just provides structural stability between the tunica intima of the capillary and the podocytes. So if a structure or ion can slip through the fenestration, or the pore, or the hole, can it also get through the interdigitation of the podocytes? And if it does, then all right, you're free to go. Okay, water, chloride ions, sodium, potassium, whatever. Red blood cells, too big. White blood cells, too big. Platelets, too big. Okay, so everything you want to hold on to can't get through this pathway. Okay, so that's why we've got multiple layers there. Okay. Again, with structure comes function. We know this throughout the body. Okay, so let's talk about each part of the nephron. So the PCT, all right, connects itself to the renal corpuscle, okay? You only find the PCTs in the cortex, right? This is where the most reabsorption happens. Okay, the most reabsorption happens in the PCT. Okay, and because reabsorption happens the most in the PCT, well, there's going to be epithelial tissue that's going to promote the most reabsorption in the PCT. So over here, okay, proximal convoluted tubule cells have microvilli. And we know that microvilli, like in the digestive system, serve as shag carpeting, right? And that increases surface area to increase reabsorption. You do not find microvilli in the DCT, in the loop of Henle, or in the collecting duct cells. None of them have microvilli, but the PCT does. Okay, so with structure comes function. Okay. All right, so in the loop of Henle, all right, we're going from one simple kind of epithelium to another. It's just cuboidal to squamous and back to cuboidal. But again, it's a single layer of cells to promote reabsorption or secretion, okay? So we have a thin segment and a thick segment. The thin segment is usually called the descending limb, okay, which you guys identified in lab. The thick limb or the thick segment is called the ascending limb, okay? Now, the descending limb or the thin segment, okay, well, what happened? Okay. Nope. So the descending limb or the thin segment, okay, is highly permeable to water, okay? Reabsorption of water happens here in this part of the loop of Henle, okay? No, not ions, just water. Reabsorption of water only, okay? In the ascending limb, it's impermeable to water, but you can reabsorb ions. Sodium, potassium, chloride, magnesium, okay? Okay, and we'll come back to this slide in a moment for the last bullet. Okay, so for this slide, I don't want you to memorize the numbers and all that. That's not the point. The point is to understand concentration and what happens. And this just kind of gives you a good story, okay? So the numbers just mean pressure, okay? And it just kind of gives you, or concentration, and it just gives you an idea of what concentration is like in addition to color as we go along the loop, okay? So in the glomerulus, the concentration just 300. Doesn't matter, we're not memorizing any of that, okay? So the filtrate is actually the same concentration as your blood. Okay, so it's the same concentrate. So it goes to the PCT, and then it starts going down the descending limb, okay, which we know reabsorbs water only, okay? So if that reabsorbs water only, all right, what does that do to the concentration of the filtrate? It makes it go up, right? It's more concentrated. So the color gets darker, okay, it gets super dark. And then you go into the ascending limb, and the ascending limb is impermeable to water, but it's permeable to solutes. So your sodiums, your chlorides, your other ions are going to leave, okay? But water's gonna stay. So what does that do to concentration? It takes it back down again. And interestingly enough, the concentration coming out of the nephron loop typically is lower than when it entered. Okay. But because the filtrate is around the same concentration as your blood, that can be a good indicator at any one point in time what your blood pressure and what your blood volume is like. Okay, but I'll repeat myself again for that. Okay, so let's go back to the previous slide here and talk about two different nephrons that we have in the body. We have cortical and we have juxtamedullary. Cortical nephrons mean that a majority of the anatomy of that nephron is in the cortex, okay? So in this picture here, the arcuate arteries and veins, that shows you that below the arcuate is what region? Medulla, and above it is the cortex. So a majority of this nephron, cortical nephron, is in the cortex. Little bit of the loop is in the medulla, okay? Juxtamedullary. Juxta means what? Like next to, right? So juxtamedullary nephrons are closer to the medulla and they have a longer loop of Henle, okay? A longer loop of Henle allows more time for the reabsorption of water. Okay. 85% of our nephrons are cortical. So a majority of our nephrons are cortical. Okay, so the remaining 15% is juxtamedullary. But that changes based on what species you are. Okay? Think about like Death Valley, Arizona or whatnot. Super arid, dry environment where you've got all kinds of iguanas and lizards and all these animals, reptiles, that can live without water for months, okay? Which we could never do. Why is that? Because a majority of those animals have juxtamedullary nephrons, so they can reabsorb massive amounts of water. And as a result, hardly urinate, because they're not really secreting much. Okay, so again, it depends on the species as to what your ratio of these guys are. Okay. All right, so moving down to the DCT. Okay, so the distal convoluted tubule doesn't have microvilli, we know that, okay? We're still simple epithelial tissue, okay? Now, we're still reabsorbing solutes here in the DCT, okay? But if we reabsorb water in the DCT, it's going to be dependent on hormones. Two different kinds of hormones that we already learned. What are they? Antidiuretic hormone and aldosterone. Yep. So, we knew from before that ADH and aldosterone secreted from the posterior pituitary, okay, targeted the kidney. That was one of the targets. Well, now we know where. The target is the DCT. Okay. It also targets the collecting duct. And whether or not you're under the influence of one or the other of these hormones will determine how much water you're going to reabsorb, right? Because if these hormones are active, then your ability to reabsorb water is going to go way up, right? Because antidiuretic hormone wants you to grab on to however much water you can. And aldosterone is similar, but it reabsorbs sodium first, right? And then water comes along for the ride. Okay. Yes, okay. So we'll talk a little bit more about the DCT and the juxtaglomerular cells in a moment. This picture I actually really like because it gives you kind of a summary of what's happening where in the nephron, okay? So in the glomerulus, in the glomerular space, in the renal corpuscle, we only have filtration, okay? That's the green arrow. In the proximal convoluted tubule, we have the maximum amount, because it's going out of the tube. What does that mean? Where's it going? It's going back into your bloodstream, right? So you have the maximum amount of reabsorption of both water and solutes, okay? Water and solutes in the PCT. That's why big arrows going out, okay? And what structure is going to pick up the water and solutes here in this location? Peritubular capillaries. Okay. All right. So then we go into the descending limb, which we know is highly reabsorbable for water. So you can see water out. Okay. Only water, not solutes, only water. Okay. Then on the flip side, we go to the ascending limb and we get the reabsorption of only solutes. Okay? And then in the distal convoluted tubule, we get reabsorption of water and solutes. Water is dependent on hormones. Okay? But then you also can get secretion from the bloodstream going into the distal convoluted tubule. Okay? And then finally in the collecting duct, you can get reabsorption of water. This is kind of like your last ditch effort to reabsorb any water should you need to. So the collecting duct is also under the influence of ADH and aldosterone. Okay? You can also secrete things from a bloodstream into the collecting duct to get rid of. Okay? And if the collecting duct is in the medulla of the kidney, then where are these secretions coming from? The vasa recta. Okay. Just like here, if we're reabsorbing, it's going to go into the vasa recta. Okay. So that's why we have two different capillary beds located in two different parts of the nephron. Okay. So the DCT here is part of what we call the juxtaglomerular apparatus. So let's talk a little bit about that. All right, so we didn't identify this on the model in lab, but this looks a heck of a lot like the model, okay? So we know that the PCT directly attaches to the glomerular renal corpuscle, to the glomerular space, capsule, and glomerulus, okay? So PCT direct connection. You've got afferent, efferent arterioles leaving, and then in the model that you identified in lab, you identified the distal convoluted tubule right here, okay? Well, in previous pictures, the DCT's over here. It's not over here, okay? So this picture is designed to just show you the general anatomy. It's not anatomically correct because the distal convoluted tubule does a drive-by in between the afferent and efferent arteriole. And it does that for a reason, okay? All right, so on the afferent arteriole, okay, we have these green cells. Those are called juxtaglomerular cells, okay? These cells are sensitive, and these cells can secrete a hormone called renin. We'll get to that in a minute. Now, at the point where the distal convoluted tubule does a drive-by in between the afferent and efferent arteriole, part of the epithelial tissue transitions into this strip of cells that's called the macula densa, only found in this drive-by area of the DCT. Okay? And the macula densa cells act as chemoreceptors. And chemoreceptors are sensitive to what? Blood chemistry, right? So we're talking about bloodstream. It's going to be sensitive to your blood chemistry, okay? Specifically your solute concentrations. So if your solute concentrations in your blood go down, your overall blood volume goes down, okay? And that's not good. Okay, so that's what these chemoreceptors are keeping an eye out for. Okay. Okay. So, on the next picture here, you don't have to memorize all this. All I want you to know is, okay, if the macula densa senses that there's low blood solute concentrations, okay, those chemoreceptors pick that up, red flag, low solute concentrations, okay, that's going to be deemed as a potential low blood pressure and or volume situation by the kidneys, okay. That's going to trigger those juxtaglomerular cells to secrete a hormone called renin, okay. Renin is going to target your adrenal cortex. It's going to tell the adrenal cortex to increase the secretion of aldosterone. Okay? And aldosterone is gonna go from the adrenal cortex to the kidneys and tell the kidneys, okay, we need to reabsorb sodium, and water's gonna come along for the ride. And the overall effect of that is to increase blood volume and thus blood pressure. Okay? So this is where the kidneys can also regulate and monitor your blood pressure based on your blood solute concentration levels. Okay? Because remember, coming out of the glomerulus, the filtrate, the concentration, is about the same as in the glomerulus. So if the solute concentration is low in the glomerulus or heading into the glomerulus, and the macula densa picks up on that, the macula densa is sensing the solute concentrations of the filtrate, right? Because the filtrate starts in the PCT, and then it goes through the nephron loop, and then it ends up in the DCT. So the DCT is still keeping an eye on filtrate concentration. And it should be relatively similar to blood concentration. And if it's not, if it's too low, that's going to trigger this series of events and for renin to be secreted in order for you to raise your blood volume and thus your blood pressure again. Okay? All right, questions about that? All right, just a quick revisit to aldosterone and antidiuretic hormone. Okay, so collecting tubules or collecting ducts. Okay, again, still simple epithelium. You can have multiple nephron loops attaching to a single collecting duct. So usually collecting ducts will have multiple branches that are attaching to it. Okay. And this is kind of your last ditch effort to reabsorb any water, okay, or get rid of any waste that you need to. Okay, because once filtrate leaves the collecting duct at the papilla, it is now urine. There's no going back. There's no modification after that. Collecting duct is the last. So the collecting duct is targeted by antidiuretic hormone. And antidiuretic hormone tells the collecting duct, reabsorb some more water, if we need to. Okay, so aldosterone is involved in the macula densa and the juxtaglomerular cells. And antidiuretic hormone is really under the influence of the distal convoluted tubule and the collecting duct. And it'll target both of those areas and increase reabsorption of water as necessary. Aldosterone will increase reabsorption of sodium and water if it's triggered to be released because you have low blood volume and or low blood pressure. Okay? Okay. So usually by the time you psychologically feel thirsty, okay, you've already been dehydrated for some time. It's just taking you a while to consciously realize that, I could use a sip of water right now. Okay. Okay, so coming on down the lane here to the ureters, okay? The ureters are going to take urine from the kidneys to the bladder. Okay, they are retroperitoneal as well, so they go posterior to the peritoneum. So their most superficial layer is adventitia. It's not a serous membrane. Okay. As far as histology of the ureter goes, it's got layers. It's got an inner mucosa, which is transitional epithelium as it approaches the bladder. Okay. And then you've got superficial to that a muscularis layer. So urine doesn't just fall from the kidneys to the bladder via gravity. You have to have smooth muscle contractions in order to propel urine to the bladder. Okay. So we again have an inner longitudinal layer and outer circular layer, similar to digestive system, except here we have a little external longitudinal layer on the outside, so think of it as like a muscle sandwich. So it's got 3 layers, okay, with a circular layer on the inside, and that just helps to move urine through the tube to the bladder. Okay. Then we hit the bladder. Okay, bladder we know on the inside is lined with transitional epithelium tissue, which allows for stretch, right? So that's the inner mucosa. There's a muscular layer in the bladder, smooth muscle only here, okay? That has, again, an inner and outer layer, middle circular layer, just like the ureters, except that muscular layer is collectively called the detrusor muscle, okay, in the bladder. So if you hear detrusor, we're talking bladder, okay? So the bladder is outside of the peritoneum, okay? So it has an outer adventitia. It doesn't have a serous membrane, but the superior surface of the bladder sits on top of the parietal peritoneum. So the inferior part of the abdominal cavity sits right on top of the bladder. So parietal peritoneum is on the superficial surface, superior surface, I should say, the superior surface of the bladder. Okay. We know the trigon or the trigony is the triangle that's made from the 2 ureters entering and the urethra exiting. Okay. And males have prostate just inferior to the bladder that females do not. Okay, so nothing terribly new here. So now in the urethra, okay, so we've gone from the bladder to the urethra, now we're switching to stratified squamous, because again, the urethra is open to the outside, so we need more layers for protection, okay? We have an internal urethral sphincter that's smooth muscle, involuntary. We have an external urethral sphincter that's skeletal muscle, okay? In females, we just have a urethra. It's posterior to the clitoris. But in males, the urethra contains 3 different names based on location, okay, which we've identified in lab. At any point in time in a man, okay, the urethra can carry semen or urine, but not both at the same time, okay? And semen is defined as sperm plus fluid. Sperm plus fluid. Okay, that's what semen is. And it's usually seminal fluid coming from the seminal vesicles that you guys have identified in lab. Okay, so the last item, again, don't memorize the pathway. We're used to these pictures by now. It's just conceptually what happens when you urinate. So micturition is the act of urination. Okay. Who's in control here? Sympathetic or parasympathetic? Parasympathetic. Okay, so this is parasympathetic control. So your bladder starts to stretch. Okay, your transitional epithelium starts to stretch, and it's going to fire off a structurally, looks like what? Unipolar, bipolar, multipolar? Unipolar. Visceral sensory neuron. Nothing new there. Okay. So that's going to pick up the sense of stretch, and that's going to send that information up to the pons. And back in nervous system, when we were talking about the pons, we briefly mentioned that the pons has bladder responsibilities, and this is it. So this tract is going to synapse up in the pons, okay, and it's going to generate an outbound motor response, okay. The motor response parasympathetically, okay, for you to urinate, all right, is to contract the detrusor muscle, okay, and relax the internal urethral sphincter, okay. Now this is a reflex. It's a reflex because you don't have conscious control over this, okay. So that's why the tract stops here at the pons. It doesn't go to the cerebral cortex. Okay? Yes, there's another synapse that's going to go up there and synapse so that you know, oh, I kind of feel like I have to go to the bathroom. Okay, fortunately. But this, contraction of the detrusor and relaxation of the internal urethral sphincter, smooth muscle, you have no control over this. Okay? So all of a sudden, you feel like you have to go. Okay? But you can pucker up, right? Because you have an external urethral sphincter that you volitionally have control over, and you can override it for a period of time, right? Until you can go to the facilities and take care of business. Okay? So that's the act of urinating. It's a reflex. So if we're sympathetic overdrive, okay, so if we're inhibiting micturition, which is the next slide, it's basically the same exact pathway, but we're just describing it in different terms. So sympathetic pathways then are going to tell the detrusor muscle to do what? If we're inhibiting urination, detrusor muscle is going to relax, okay? Internal urethral sphincter is going to contract, okay? Yep. So most of the time, your internal urethral sphincter is contracted. Okay? It's only when you have to urinate that it will reflexively relax. Okay? Okay. That's all we need to know about urinating. Questions about urinary system?